Golgi apparatus and a brief overview of what is called as vesicular trafficking. So Golgi apparatus is again an integral part of the endomembrane system, and it plays an important role in sorting cargo that has to be delivered to different parts of the cell. So let us look at the learning outcomes of the session. So Golgi apparatus is a stack of flattened sacs of cisternae. So this is something that is very similar to endoplasmic reticulum, uh, but they are made up of cisternae that typically are 2 to 6 cisternae and independent of each other. But depending on the function of the cell, the stack size can vary. It can be as big as 20 stacks as well. The Golgi apparatus is observed to have interconnected tubular structures at the cis and the trans phase especially, and such networks that are formed are called a cis Golgi network and the trans Golgi network. In the Golgi apparatus, proteins coming from the endoplasmic reticulum are processed and they are packaged, and then they are sorted into different vesicles, and those vesicles are deported to different destinations. Thus, it is involved in what is termed as protein sorting. Glycosylation and processing of oligosaccharides in the different compartments of the Golgi are significant for the final quality of the biomolecules formed. Now, in this, you must note that although protein sorting is something that has been extensively studied and reported, one has also found that it is the lipids and the proteoglycans also that are either transported to different organelles or transported to different parts of the cell. For example, especially the plasma membrane. So if you look at the structure of the Golgi, you can see that they are made of flattened sacs, and these flattened sacs are what is called as cisternae. But towards the cis side and towards the trans phase, what is the cis phase? The phase that is the region where, or the side of the Golgi apparatus that is facing the nucleus is what is called as the cis phase, and the side of the Golgi apparatus that is facing the plasma membrane is what is called as the trans phase. Now you would see that many Golgi can come together through interconnected vesicles. This is also something, so you would consider then this as a whole Golgi complex, or you can have as small apparatus as just 2 cisternae. Those cisternae that are close to the cis Golgi network, so you can see that at the cis Golgi network you have tubules that are interconnected, whereas these are flattened sacs. So again you will find tubules present at the trans Golgi network as well. So this is very distinctively two faces of the Golgi apparatus. The cisternae that is close to the cis Golgi network is what is termed as the cis cisternae. The cisternae that are close to the trans Golgi network are termed as trans cisternae, and what is between the cis and the trans are what are called as the medial cisternae. So what is very clear is that the Golgi apparatus is compartmentalized. Well, this is one compartment, this is the second compartment, this is the third compartment likewise. And interestingly, each compartment can have defined set of functions. Now, when you have proteins and lipids arriving at the cis phase, they undergo changes as they move through the Golgi, and having undergone those changes at the trans phase, they are sorted and then transported to different parts of the cell. Some may go to the lysosomes, some may go to the plasma membrane, and likewise. So this sorting that is done with every vesicle leaving the trans phase, there is an address or there is a tag on these molecules that are suggestive of the fact that which proteins are sorted to which particular destination. So that is something that is very important with respect to Golgi, that it not only trims, cuts, adds sugars to the different proteins, they also kind of package it into vesicles, and then from there they are dispatched to different parts. So therefore, one also calls the Golgi apparatus as a shipping department of the cell, because it it takes up cargo, it processes the cargo, packages the cargo, and then dispatches it to different routes. The Golgi apparatus, therefore, is also able to do a further quality check of the proteins coming from the ER. So we already know that ER takes care of quality check. It ensures that the protein is folding properly. But in the Golgi, what can be done is that the proteins can be further undergoing modifications, mainly with respect to the oligosaccharides. So you could have sugars being added, or you could have sugars being removed, or you could have other chemical modifications being done. And these changes are actually improving the function of the protein, and thereby the quality of the protein is improved. Now, the processing within the Golgi apparatus generates an address for every protein. So, what we mean by that is that suppose you have a protein that is phosphorylated at, say, a particular residue or at, say, a particular sugar, then that phosphorylated sugar becomes a signature for that protein. So, depending on that signature tag, the protein is sorted into a vesicle, and that vesicle will carry this protein only to a particular destination. So you can see how the protein itself now carries an address, and reading that address, the protein is dispatched and given to a target area. So that is something that the Golgi takes care of with a lot of accuracy. Now, to look at what kind of processing happens in the Golgi, we are just taking a few examples. So, for example, when you have proteins coming in from the ER at the cis Golgi network, what happens is sorting. What kind of sorting? So, here the sorting that is done is, say, for example, from the ER, you have vesicles coming in carrying different proteins. Now, suppose the vesicle also carries a protein that actually had to remain in the ER. So that means that protein would have an ER retention signal. So in the cis Golgi network or cis cisternae are present certain receptors that are called as KDEL receptors, because KDEL is the ER retention signal. So all proteins that have KDEL will bind to the KDEL receptors. Those receptors will again bud off from here and be sent back to the ER. So such kind of sorting is done in the cis phase of the Golgi apparatus. Also, what is done is that the sorting is also done in such a way as to what the protein has to undergo what kind of changes. So as you can see over here, in the first part of the Golgi, the phosphorylation of oligosaccharides is carried out, and that too specifically on lysosomal proteins. In the second cisternae, you can see you have removal of mannose. In the third cisternae, you can have removal of mannose, addition of N-acetyl glucosamine. Then you have addition of galactose, addition of sialic acid. Then you have sulfation of tyrosines and carbohydrates. So you can see that every compartment has a set defined function that is being taken care of, and as the proteins move from the cis phase to the trans, wherever it has to be modified, it gets modified. And based on what are the final modifications, those modified proteins or those modified cargo proteins or cargo are then sorted into different vesicles, and those vesicles and then are sent to either the lysosome, or they become the secretory vesicle, or they go and become part of the plasma membrane. So this is what happens in the Golgi. And interestingly, what has been observed is that most of the enzymes that carry out these functions are membrane-bound enzymes. So they are found in the membranes of the Golgi apparatus. Also interesting to note over here is that the Golgi is able to carry out what is called as O-linked glycosylation. Now, this is highly significant because we know that certain compound proteins like proteoglycans have what is called as a core protein, and to several of its serine residues is attached what is called as glycosaminoglycans. So, glycosaminoglycans are disaccharide units that are repeated. So those sugars are attached to the serine OH of a core protein, and that is basically what is O-linked glycosylation, and that O-linked glycosylation happens in the Golgi apparatus. So you can see how glycoproteins are also formed and proteoglycans are also formed in the Golgi. So basically these specific additions or deletions or trimming etc. results in a particular tag being formed, and that tag enables sorting. Okay, so for example, this is, say for example, a core oligosaccharide that is already bound to a protein. Okay, and now this core oligosaccharide is undergoing processing where addition of different sugars take place to form what is called as complex oligosaccharides. Or say for example, you have another common type, which is that you have high mannose oligosaccharides being formed where several mannoses are added to form a moiety which has a high mannose content. So the products of the carbohydrate modifications carried out in the Golgi are quite complex, and this leads to the field of study that is called as glycobiology. In fact, many people have looked at glycobiology to understand the functioning of different proteins, to understand how cells communicate with each other, to understand how cells interact and bind to each other. All these different things are related with the different glycobiology. And that glycobiology is mainly associated with the changes that happen in glycosylation in the Golgi. So this is just depiction of the same. You can see that in the endoplasmic reticulum, what was added as a oligosaccharide moiety had 14 sugar residues with 2 N-acetyl glucosamine, 9 mannoses and 3 glucose. Now within the ER itself, this sugar moiety can be trimmed down or can be altered, and then the altered protein with the oligosaccharide is transferred to the Golgi. So at this stage, what you have is a high mannose oligosaccharide. Now that high mannose oligosaccharide can further be trimmed, or you can have phosphates added to this, etc., etc., etc. And with every cistern, what happens is that you can have a complex oligosaccharide being formed. And in this complex oligosaccharide, you can see that when NaNA, that is N-acetyl-nuramidinic acid, is added, which is also called as sialic acid, is added, you can see sialic acid is negatively charged. So therefore, the sugar moiety also attributes a negative charge to the protein, and that has an important or has a significant role. So both proteoglycans and glycoproteins are formed in the Golgi, and these glycosylations not just help in sorting, but they also are helpful in cell-cell interactions, binding to lectins, enabling formation of gel-like environment. For example, mucus. So there is mucin that is formed, that is present in the mucus, and we all know that mucus is generally a mechanism of defense where because it is sticky, it holds on to a lot of germs, and that way it becomes the first line of defense for our immune system. Now, what has been understood or what has been tried to explain by various models is that what happens to a vesicle or what happens to a cargo protein as it comes from the ER to the Golgi. So eventually in the Golgi, how does the processing happen? So one of the model that has been contemplated was something that is called a cisternal maturation model. So as the word itself suggests, what happens is that the cisternae that is formed over here matures into the next cisternae, that matures into the next, that matures into the next, and finally at the trans end, the matured cisternae get again disrupted into small vesicles, which then move to different destinations. So this becomes maturation model because every new cisternae is slowly moving one place, okay, getting more and more mature. So here what you see over here is a cluster, a tubular cluster that is formed because of various vesicles fusing to form this. Now this will become the cis Golgi network that is going to get mature to form the cis cisternae, which is going to get mature to form the medial cisternae, which is going to get mature to form the trans, and from the trans, as the trans Golgi network, you will have the vesicles going out. So this is one model. There have been some experiments that have showed that this is possible, but there is another model also which is called as vesicle transport model. So in this case, what is being said is that any vesicle that comes from the ER, of course, becomes what is called as a vesicular tubular cluster, which is also called as endoplasmic reticulum Golgi intermediate compartment. So it's almost like an organelle in itself that forms the cis Golgi network. But from the cis Golgi network, whatever protein has to be further processed is actually budding out as a vesicle, then budding in over here, or rather endocytosing here. Whatever changes has to happen over here to the protein happens, then it gets again exocytosed out, then endocytosed, exocytosed, endocytosed. So here the cisternae are not maturing, but the protein that gets modified is just coming out as vesicle and then again taken in as vesicle. That way, exocytosis and endocytosis is leading to finally the trans Golgi network having vesicles with specific processed proteins that can be delivered to different destinations. So this is what is called as vesicle transport model. Many experiments have shown that this is definitely the case, and even through electron microscopes, it has been found that Golgi apparatus always seems to be, surrounded by a lot of vesicles. So that kind of, gels with this model per se. So in this case, new cisternae will form due to vesicles fusing with each other, and every cisternae then will mature and at the trans phase will bud off as vesicles. The proteins that are processed in the first cisternae bud off to the next for further trimming, and that is how at the trans phase finally you will have the sorting done. So this and this model, there are some experiments that say that cisternal maturation model is what is happening, and some that say that vesicle transport model is what is happening. There are more experiments that are kind of believing in this model, but one is not really completely sure. Now, interesting to notice that these Golgi are positioned in the plasma membrane by microtubules that are the cytoskeleton. Now, therefore, there has to be something to which the cytoskeleton can bind to in the Golgi. So what has been found is that on the surface of the Golgi are several specific proteins. Here what the name is given is Golgines. These Golgines can bind to cytoskeletal interactor domain, and then the cytoskeleton will bind to it. So when the cytoskeleton is formed and it is binding to Golgi, it will hold the Golgi in place. Secondly, what has been found is that these molecules that are present on the surface of the Golgi are able to interact with several regulatory proteins, including RAB protein, which is a GTP binding, which is a G protein. So it can bind to GTP, and we know that when it is bound to GTP, it is having a particular function. When it is hydrolyzed to GTP, it can have a totally different function. So therefore, there is a regulation because of GTP and GTP hydrolysis into GDP. Also, what has been understood is that these Golgins also assist in the vesicles to move from one cisternae to other cisternae, as these can bind to proteins present on the surface of the transport vesicle. So Golgi are positioned due to microtubules, one, but they also are positioned due to the proteins that are present in the Golgi matrix, such as the Golgins. Now we come to a concept that is so very integral a part of Golgi, or rather, Golgi becomes a central part of the vesicular trafficking. And vesicular trafficking today has been very well studied. The molecular mechanisms are highly defined, although more is to be known, and it is beyond the purview of this session, but it is a very important phenomenon and interesting phenomenon, and therefore including it with Golgi apparatus in this session. So as you can see, this is the endomembrane network. So it begins right from the nuclear membrane to the ER, to the Golgi, to lysosomes, endosomes, etc., to plasma membrane. So right from the nucleus to the plasma membrane, you can have cargo proteins being transported from one end to the other end. So you have what is called as vesicles moving from inside the cell from the nucleus onwards to the plasma membrane. So that movement is what is called as anterograde movement. And you can have at the same time molecules from outside the cell taken in through endocytosis or from the membrane in or from different parts of the cell intracellular compartments. They can be taken, and those molecules can be transported in the form of vesicles from the plasma membrane to the Golgi, from the Golgi to the ER. So this traffic is what is called as the retrograde movement. So you can have two-way traffic definitely, uh, but the vesicles that are trafficking these the proteins that are vesicles that are trafficking these cargo proteins can be different. So here, as you can see, all vesicles that bud off from the ER are due to proteins that are called as COP2 proteins. So these COP2 coating proteins form a coat to form a vesicle, and then these vesicles are transported to the Golgi. So the COP2 is always um, a transport vesicle from the ER to the Golgi. But then you can also have from Golgi vesicles coming to ER, and those vesicles are coated by COP1 proteins. So COP1 coated vesicles are transporting proteins from the Golgi to the endoplasmic reticulum. So it's very clear. COP2 goes from ER to Golgi, while COP1 vesicles go from Golgi to ER. So this is only one way direction, and COP1 is also one way direction. So they are basically sharing the load. So whatever has to go from ER to Golgi is taken by COP2, and whatever has to go from Golgi to ER is taken by COP1 proteins. This that you very typically see is the endoplasmic reticulum Golgi intermediate compartment. So this is formed before it becomes part of the Golgi apparatus per se. Now, this was one phase, the cis phase of the Golgi apparatus. But from the trans phase of the Golgi apparatus, the vesicles that are budding out, they are budding out because of clathrin. So clathrin is another set of proteins that can coat vesicles. So they are responsible for the exocytosis. So they are called as clathrin coated vesicles. So to and fro from the trans-Golgi network to the plasma membrane, or trans-Golgi network to the lysosome, or trans-Golgi network to the endosome, these are all because of clathrin coated vesicles. So this phase of vesicular trafficking is carried out by clathrin coated vesicles, while this phase of the vesicular trafficking is controlled by COP2 and COP1 coated vesicles. So that is what we understand by vesicular trafficking, and this has been extremely well studied. People know till the depth of how the COP2 forms the COP2 vesicles, how COP1 forms the COP1 vesicles. To that extent, the molecular mechanism is understood, and also how these COP2 coated vesicles actually bind to the cargo protein, and within the vesicle the cargo protein is carried. So everything or many of these things are known. Now, one important fact about vesicular trafficking, that it is clearly a process of exocytosis and endocytosis, or what is called as budding off of vesicles and docking of the vesicles and fusing of the vesicles. So it is basically budding off and fusion. That is what is happening. So as you can see over here, and there are some specific proteins that help in the process. So I would just name one or two of them. So for example, say this is an this is endoplasmic reticulum, and this is a soluble cargo that has folded properly. So this cargo will have will have probably a stretch of amino acids that will be recognized by certain transmembrane cargo proteins. So you will have interaction between the two, and then these will accumulate in a region which is budding off um, as a vesicle. So you would have the coat protein coming and binding, and this coat protein that comes and binds help forming the vesicle, and here it will pinch off. So now you have the complete vesicle being formed, but you can see that the vesicle has the cargo within it. Once the vesicle has been formed, the role of the coat protein is done. It had to bud out the vesicle, which it has done. So now the coat protein will again shell off itself, and these coat proteins can be reused to form another vesicle. Now this vesicle that is carrying the cargo will have to go and attach to the target membrane, and so that is helped by this transmembrane protein, you see. So what happens is that this will tether to a tethering factor with rab acting as a regulatory protein. Rab is a G protein. So when you have the tethering of the vesicle done, then you have the vesicular snare. So here you have a vesicular snare. This is a protein that can bind to target snare. So you see, this will recognize the target snare, and so you would have the V snare binding to T snare to dock. So you have the vesicle now docking on this membrane, and once it docks on the membrane, a molecular mechanism follows up to fuse the vesicle in, and you can see over here how the cargo protein which is present here now is present over here. So this is what is the vesicular trafficking, and you basically understand that here you have budding off of the vesicle, and here you have fusion of the vesicle with the target membrane. So this is how transport of cargo is carried out through vesicles. So let us make the conclusions. The Golgi apparatus is organized into a stack of flattened sacs of cisternae with a network of tubules at both its cis and trans faces where sorting takes place. Each cisternae has assigned specific processing enzymes that are able to process the cargo coming from endoplasmic reticulum further to enable its sorting to different destinations. One of the major functions of the Golgi apparatus is in glycosylation, either modifying the N-glycosylated oligosaccharides or by adding glycosyl groups by O-linked glycosylation. All transport to and fro Golgi apparatus is by vesicles, which makes endocytosis and exocytosis a way of functioning for Golgi, leading to what is called as vesicular trafficking. So the Golgi apparatus, also called the shipping department, processes and packages the cargo such that each cargo has an address that defines where it has to be delivered to, and the processing ensures perfect quality of the product delivered. Thank you.